Thanks. This is going to be the last paper 3 review that I do, and then I would have covered all the new papers, other than the specimen paper, but I would have covered all the new papers in the syllabus. All right, and this is October, November. This is the most recent test that they've published until tomorrow. 2022 and paper 3.1, it is the same as 3.2 and 3.3. They did not have any other versions. All right, and again, you can download this on your own and then do it or print it out. All right, so this is chapter 6, doing molecule or particle transport. And so it starts with there's continuous movement of substances, orange is better, through cell membranes of cells of marine organisms, and substances move from cell to cell or are exchanged with the surrounding seawater like gases or waste. And you see 3 processes by which substances will pass through a cell membrane. Complete the table by placing a tick in the correct boxes. And it's nice that they now supply what a tick means, because I know in America we don't say the word tick, we would just say do a check mark. So the processes are diffusion, facilitated diffusion or helped diffusion, and active transport. And then we need to decide if they are moving from a higher to lower concentration, if it's a passive process, which means it does not require ATP, and if glucose uptake is an example of it. All right, so for the first part, diffusion is moving from a higher to lower concentration. That literally is what it is. So you have a high amount of something moving to a lower amount of something. All right, and additionally, the example I use in class is if your car was like on the top of a hill, so like the high amount, and then it went to like lower elevation, would that require you to turn on the car? And no, it wouldn't. You wouldn't have to use any gas. It could roll on its own. It doesn't require energy. So this is also considered a passive process. And again, this means no ATP. Not used for glucose uptake. Glucose is much too big. So diffusion is like just for like gases coming into and out of the cell. Things that are small. Facilitated diffusion. This is like helped diffusion. And this can be with a channel protein or a carrier protein. Channel proteins only do facilitated diffusion or a passive transport, so high to low, and then carrier proteins can do both. They can do active transport, so going from a low amount to a high amount, and they can also do passive transport, a high amount to a low amount of something. So facilitated diffusion can and does, because it's right in the word diffusion, can move things from a higher concentration to a lower concentration. This is also a passive transport or passive process, and it can be used for glucose uptake. If that's the case, we'd probably use a carrier protein so it could fit through the cell membrane, because it's much too big to just do it on its own. Active transport does not move something from a higher concentration to a lower concentration. Active transport moves things from a lower concentration to a higher concentration. And in the same analogy of your car, if you're moving your car from a low elevation to a high elevation, you would need to turn it on, or up a hill, you would need to turn it on, use energy, use gas, and so that requires energy. So we're not going to do a tick mark in the first box. It is not a passive process. It does require ATP, and we can use it for glucose uptake. And again, this would be when we would use a carrier protein, and ATP would be required. Our muscle cells do this. Our quads, we pump so much glucose into the cells that would fuel our quads in our body, so that if we are needing to run really quick or like quick spurts of energy, we're not waiting for our blood to send glucose over. We already have extra stores of it. So we are purposely putting a lot of glucose into our muscle cells when maybe there's not that much glucose floating around or moving around within our blood. So osmosis, this next one, involves movement of particles by diffusion. Explain why osmosis is sometimes described as a special case of diffusion. And this is because of H2 osmosis. Osmosis only implies that water is being moved. Osmosis only applies to the movement of water. And that's MVMNT, movement. Again, tomorrow we're not going to be using any sort of abbreviations. Here we see a plant cell and then an animal cell under normal conditions or normal like aquatic conditions. And they are identifying your cell wall, your cell membrane, and the cytoplasm. Okay. And they want on the next page, it says sketch diagrams to show how the plant cell and the animal cell would look after being placed in a concentrated sugar solution for 5 hours. And we're going to want to include just basics of what they have here, which is going to be the cell wall for the plant, cell membrane for both, and then essentially what the cytoplasm would look like. But we're not going to use any shading. In mine, I also did include the nucleus for both of them, just where that would be. So to help you kind of imagine what this is discussing, so here's our sugar solution. Sugar is sweet. I'll make this pink. All right, the sugar solution, and then inside we're going to have our cell. Could be plant or animal. I'm just going to do an animal right now. And kind of like how we started in class. So outside, we want to show water potential. So we have a high amount of sugar. And so we're going to have the inverse for water, low water potential. And the opposite will be true for inside the cell. We're going to have comparatively, and this is always comparing the cell to the solution that it's in, whether we're not even talking about a cell, we're talking about an entire organism, like a fish going into a different type of salinity, or as simple as the visking tubing practical, where we put different concentrations, the visking tubing in different concentrations of water, sugar water in the beaker. Maybe in your biology class, you did this with eggs. Okay, so comparatively, we're going to have a lower sugar solution, lower sugar, and a higher water potential, or more fresh water, comparatively. And water always moves through osmosis, so it moves from an area of high water potential to an area of low water potential to try and be in equilibrium. And that's just the way the particles move. So no matter if this was a plant or an animal cell, water will be lost. It's leaving the cell. And this is what we do when we gargle salt water on purpose to try and alleviate the pressure that's in our cells. And then, of course, we're going to spit it out. Or if we have swollen joints or swollen muscles, we'll sit or soak them in like an Epsom salt solution, just trying to make a different concentration gradient so that all that swelling or water held up in our tissues will relieve itself. So in order to do this, we want to kind of look at what they were giving us and build from that. So the plant cell has a cell wall. And I would do this in pencil. And of course I can cheat. Okay, and here's the edge of the cell wall. All right, and then we're going to want to show that like some plasmolysis has occurred, or like breaking down of the cytoplasm that's contained within the cell membrane. And so this will look something like this. We're showing the cell membrane has kind of like pulled itself off from the cell wall. And then my nucleus. And I didn't shade it or anything like that, but notice the cell membrane is definitely pulled itself off from the cell wall versus here you can't see, it's all the cytoplasm is full in the background. And the trigger pressure is high. Animal cell, We don't have a cell wall. So instead of it being nice and round, it's going to look more like it's shrinked, shrunk. So instead of it being nice and plump, nucleus. Label the cell membrane on both of the cells you've drawn. When you're labeling, you're going to want to use a pencil for your label line and a ruler. And then all of the label lines are written in ink horizontally. And again, this would be something you could absolutely use a pencil for. All right, and then we need to kind of explain what we wrote. So explain the changes in the plant and the animal cell when it was placed in the concentrated sugar solution. Use the term water potential in your answer. And so sometimes I'll just like dumb it down for myself in my class and say water goes from where it's more fresh to where it's not as fresh. Now we're going to change the word from more fresh to higher water potential, and then not as fresh to lower water potential. So water moves or diffuses from an area of high water potential. I'm just going to put water pot, and this would be inside their cells, to an area of low water potential, and this was in the sugar solution. All right, and then we need to say, explain the changes in the plant cell and in the animal cell. In the plant cell, your cell membrane pulls away from the cell wall. And in the animal cell, and this is the first video of mine that you're seeing, I show each different mark from the mark scheme with a bullet point, and I'll indent whenever they're like kind of going to further detail about the one that was previous to it. And actually it'll make better sense if I get rid of the word in. I'm just going to say the animal cell shrinks. Or you could even say the cell membrane shrivels. Okay, so I've already done this question in class, so I just included the answers that I've already done, and then I'll go over it too. Okay, so this is showing chemosynthesis, which is 7.2. We're looking at part of the food web there. And of course, we have vent bacteria and endoriftia, which lives inside of riftia. They have a mutualistic relationship. They're at the base of your food web, so they are the producers. Even though it says bacteria, it is like a it's a chemosynthetic bacteria, but it is the producer there. You can tell because it is on the bottom trophic level. Vent bacteria can form large mats, like you see here, drawn on this hydrothermal vent. Or you have the chemosynthetic endoriftia. Endo means inside, and it lives inside of this riftia tube worm. Similar to corals, and so xanthelle lives inside of the coral and does photosynthesis. This is endoriftia, lives inside of riftia. And again, endo means inside. And the exam question, describe how vent bacteria and endoriftia carry out chemosynthesis. So what does vent bacteria do and what does endoriftia do? They're both producers. They use inorganic compounds. If this was photosynthesis, we would say they use water and carbon dioxide, and with the energy from the sun, they synthesize glucose. So they use inorganic compounds. We can give an example. hydrogen sulfide, carbon dioxide, also iron. What else? There's also methane, some hydrogen. Okay, and this H2S is the energy source. So it's similar to where we would refer to the sun as being the energy source in photosynthesis. This is chemo or chemical synthesis, synthesizing things or making things with chemicals, dissolved chemicals. Okay, and so that's the energy source they use. And they fix or convert carbon, similar in photosynthesis. It is carbon fixation. We're converting it from one form to another. And this is to produce chemical energy or energy for the organisms into glucose. Oses are sugars. You can call them carbohydrates, or you can call it organic material. Glucose is a chemical, which is why we say they are producing chemical energy. Glucose is a chemical, and energy is stored in the carbon bonds that hold it together. And you have 1, 2, 3, 4, 5, 6, which is why it's C6H12O6. All right, describe, so what the relationship between endoriftia and riftia. All right, and then again inside main portion of the riftia tube worm is a tube worm. This is its calcium carbonate shell. The internal side is called the trophosome. Inside it, you have endoriftia. Lives inside of it. And the job of riftia is essentially, we're not going to use this as an exam answer, but it's like to act as a straw. And it takes in your inorganic molecules or compounds. So hydrogen sulfide, carbon dioxide, it's going to deliver or transport these chemicals that are coming out of this hydrothermal vent, and it's going to the endoriftia that is inside of riftia. Now, if it's vent bacteria, they don't need riftia tube worm to do this. That just absorbs it from its environment. And so to describe the relationship, they have a symbiotic or mutualistic relationship, mutualism, because they both benefit each other from this relationship. Endoriftia, endo again means inside, lives inside riftia. Endoriftia, identify that. Endoriftia provides organic molecules or organic material or glucose or carbohydrates for the riftia tube worm. This is how it gets its energy. And then how do they do this? So riftia, the tube worm, transports or absorbs and then provides the inorganic molecules like carbon dioxide, hydrogen sulfide to endoriftia producer bacteria. It is chemosynthetic bacteria that lives inside of it. All right, vent octopus and vent ratfish are both top predators. Use the figure to explain, so give reasons, why food chains involving the vent octopus are more energy efficient, or you get more energy from it, than the food chain involving the ratfish. It's a very AS level question too. All right, so they're talking about these up here, vent octopus and vent ratfish. And so if I'm looking at the vent ratfish food chain, we have trophic level 1 is the vent bacteria, trophic level 2, the vent amphipod, trophic level 3, the Galatheid crab, and trophic level 4, your vent ratfish. Now the vent octopus, trophic level 1, the producer always at the bottom, endoriftia, lives inside of riftia. And then we can go vent mussels, trophic level 2, vent octopus, then is trophic level 3. Or vent endoriftia to riftia, trophic level 2, and again, vent octopus, trophic level 3, again, Or if we follow the arrows that point to it, we have vent bacteria, vent shrimp, trophic level 2, and then again back to the vent octopus, trophic level 3 again. So vent octopus is staying at trophic level 3, where the vent ratfish is trophic level 4. And you should remember from AS Marine and then regular biology class that every trophic level you go up, you only gain about 10% of the energy. So being in trophic level 3, there's going to be more energy remaining, hence why they refer to it as energy efficient, than being in trophic level 4. So if an octopus has 3 TLs, trophic levels, we're not going to use that tomorrow. Or we could say the inverse, the Ventratfish has 4 trophic levels. Only about 10% of energy, and RG, again, abbreviation that we're not going to use, is transferred to the next trophic level, TL, where we can say that there's more energy lost with the ratfish for that same reason. All right, an oil seep is a natural escape of oil and gas, such as methane. Methane is CH4. Up through the seabed, oil seeps account for almost half of the oil released into the ocean every year. Natural oil seeps release oil slowly over time, and this will allow ecosystems to adapt because it's slowly over time, and that's when they're natural. Ecosystems around oil seeps are very similar to those shown in figure 2.1, which is this. And so they're using the chemicals from the oil seep to synthesize organic material. Drilling companies use oil seeps as indicators for possible large oil deposits. Explain, so why and how, give reasons. Construction of an oil drilling platform would affect the water quality. So we're going to talk about water quality with reference to the impact on the community of marine organisms in that immediate area. So we're going to answer about here's what's going to happen to the water quality, and then we need to say what's going to happen to the marine organisms in the area surrounding it. So we can talk about the sediment that's going to be released from the construction, as I talked about, explain how the construction, and what it's going to do to organisms. So it can block the gills of fish. It can cause the water to be very turbid, and then they won't be able to locate any food. It can smother your benthic organisms. And I'm going to assume that we're not going to go with this is going to block sunlight, because your photic zone is about 150, max 200 meters in the ocean. And I'm going to assume this oil seep being at the seabed is not where photosynthesis will be occurring. Also considering they said very similar to the figure shown in 2.1, and this is chemicals. So this is not using energy from the sun. So the first thing I'm going to talk about is the sediment release. Again, from the construction. Block the gills of fish. And then it's, it's not on the mark scheme, but you could totally go into that would cause less surface area for them to extract oxygen, and it's going to inhibit cellular respiration or aerobic cellular respiration. They won't be able to grow as much or be able to metabolize food as much, and be able to swim as fast because they won't be getting as much ATP from respiration. We could smother when this sediment settles. Benthic ORG organisms. Additionally, it could cover, whenever it settles, the vent bacteria that would be doing this chemosynthesis process. fish can't see, and so therefore they can't find food. And this cover or smother vent bacteria. Actually, should go right here. Let me get rid of that G. There's more. So releasing oil or drilling into the sediment could obviously release toxins that can poison fish, or it could bioaccumulate. And before I say that, I should finish out this cover the vent bacteria. That would, if it could, the release of the sediment could cover vent bacteria again, and that is going to cause less of a food source for consumers, because those are your producers. One second, my daughter's coming. What's going on, baby girl?
And do I look play on the swings with me?
But I can't watch you outside right now because I can't pause this.
Well, I mean, we will be fine out there.
No, somebody has to be out there with you, okay? You can fast forward this or just pause it or something. Would you ask Jeremy, please?
Hi.
Give me 20 minutes. I already told her, Daddy, what's up? Sorry, okay. Okay, or can accumulate in organisms, bioaccumulate. You bioaccumulate in the organism, you biomagnify up trophic levels. Or just like a general mark that says that drilling could increase the oil that's released and the community can't adapt. So that's looking back at here where it says natural oil seeps release oil slowly over time. And this allows ecosystems to adapt. And again, they want you to talk about the community, which is all of the organisms there. Everything that's alive is the community, where the ecosystem adds in everything that's alive and the abiotic aspects. But at the rate that this new oil could be released from drilling into it, they may not be able to adapt. And all in all, I think this paper, this October-November paper, is really easy. And the community can't adapt. Methane, again, that's CH4. In the atmosphere, contributes to the enhanced greenhouse effect. The seabed is an important sink for methane gas. Few of the small bubbles that escape from methane gas seeps reach the ocean surface. So few of the small bubbles that escape actually reach the ocean surface. So explain why some scientists are concerned that drilling for methane at gas seeps would affect climate change. They essentially told you the answer. We kind of just need to give it some synonyms. They said that methane will contribute to the enhanced greenhouse effect. And so this will cause more methane to escape. We already have few bubbles escape, and they said of the few that escape, or those some that escape, few of them actually reach the surface. So we're going to encourage more to be reaching the surface. And rise to surface. Okay, get a mark for mentioning that it's going to go into the atmosphere. Causing more heat to be trapped. And we know this because they said it contributes to the enhanced greenhouse effect. And this is 9.2, global warming, greenhouse effect. Or you can refer to this as long wave radiation. Specifically like infrared light, which it has long wavelengths. And further contribute to the increase in global temperatures, or you can say further contribute to global warming. All right, now we have some life cycles of marine organisms. Marine mussels can tolerate wide range of salinities. State the term for organisms that can tolerate wide range of salinities. Wide prefix is uri. Salinity is haline. Uri-haline. The opposite of this is stenohaline. All right, we're looking at the life cycle of a marine mussel. They have a complex life cycle. And use the figure to identify two features which show that the marine mussels have a complex life cycle. All right, so if we're starting at the adult mussels and they have spawning, we have several different free-floating larval stages. That's one. Metamorphosis, that's another. Metamorphosis is like the changing of like their form. So like an infant, even though they're going to grow, they always look like they belong to a human. We actually have a simple life cycle. These organisms have complex life cycles so that they can inhabit different environments. They don't cannibalize each other. There's also different food sources. They, again, different habitats, and they look totally different. And it's not going to be mussels. Small mussels called spats will settle on the bed, or juvenile mussels will grow attached to the seabed. It's not that. It's going to be what I just put exclamation points by. So there's several larval stages, and this is straight from your syllabus, to recognize complex life cycles with metamorphosis and several larval stages. Identify a stage that is non-sessile, which means sessile means that it can't move. And so this is saying identify a life cycle that can move. If it's sessile, it's like sedentary. And this is going to be your free-floating larval stages, because here settling on the seabed, here they're growing attached. Adult mussels are still attached. So this would be like the trochophore or the veliger stages. Mussel aquaculture takes place in temperate waters. Mainly in Europe or North America, growers either collect spat from natural mussel beds on the seabed or use hatchery-produced spats. Natural mussel beds are important feeding areas for ducks and other wild birds. Suggest two advantages and two disadvantages of producing spats in a hatchery instead of collecting spats from the seabed. Okay, so if you're producing them in a hatchery, you have a guaranteed supply. So you'll never have to worry about climate effects or environmental effects limiting your supply. This doesn't decrease the food supply for seabirds, as they referenced, or seabirds, wild birds. Coming from that right there. You can also say the spat is protected from adverse weather. But wait, there's more. Maybe algal blooms. Predation. I'm going to spell it right. And with this, it's toxins from algal blooms. Some of them could be harmful algal blooms. Protected from, and I should say, start with that, protected from, or they don't have to be around, high temperature due to climate change. And they did also allow AVP, which means alternative valid point. And I won't write it, but I also said they're protected from potential desiccation, so drying out. Some disadvantages. Now having them, so this is a disadvantage having to grow in a hatchery. It's expensive. So there's high cost. And again, that's an American dollar sign, so you're not going to use that on this exam. It is an international exam. To setting up the hatchery. You know, that's like your initial investment, but your day-to-day is also expensive. So the maintenance, the daily running for the water pump, the oxygen pump, filtration for waste. High running costs. You could also say the same mark would talk about electricity to maintain the temperature, maybe keep some lights on, artificial feeding, which actually will be, the feeding is actually one in of itself. Filters, filter out the waste. And then there's artificial feeding, where they would be fed from the natural water cycle if they were in their natural environment. Artificial feeding is required. And those are things you'd have to hire people for as well, and they would have to be knowledgeable on how to do it, where the ocean could maybe just do it for them. Okay, I actually have this answer on the next slide because was a lot you could write about it. But one method used for mussel aquaculture involves wooden poles, which are attached to the muddy seabed in rows. Coconut fiber ropes is wrapped with a spiral around the poles, and mussels attach themselves to the rope by threads from the shell. And that gives them like a rough surface to attach to. So you can see all the mussels here. It also allows them to like take this long rope and maybe like unwind them. And they're attached to the coconut fiber, so they're not, you don't have to remove the entire wooden pole. You just remove that one strap. This is called the Boucher method. And your question is, the mussels are grown in intertidal areas, so you're going to have influence of the tides. That's going to help with maintaining the water temperature, or like bringing it back down whenever the tide comes back in. So also help reoxygenate the water. This will help maintain the salinity. So maybe if there's too much evaporation and then its salinity got too high, so this will help with that. It'll help bring in food. Shallow bays and estuaries. Estuaries have a lot of nutrients there, so that could be helpful. They're left to 12 to 15 months for harvesting. And so they suggest, so give an idea and explain, so back up your idea, give a reason why you said that. 2 advantages, and give an idea and explain your idea of 2 disadvantages of growing mussels in the intertidal areas and within estuaries instead of offshore cages on the seabed. So what's your idea and why of how this could be good to grow them in an estuary or in shallow bays instead of offshore cages, so in the open ocean kind of? And then 2 disadvantages for growing them at the estuary versus offshore. And it suggests and explains. So you're going to have your idea and then your rationale for it. Okay. So I already done this one. So for advantages, we're going to be able to check for predators easier, to have it in a shallow intertidal estuarine area. We can also be checking for biofouling. There's regular flow of water with food sources in it. and regular flow of water that will remove waste, a regular flow of water that will maintain the oxygen. And on the side here, there's protection from storms and waves being in this intertidal area versus offshore. Those waves can get really big. You don't need a big boat. Where going offshore, you would need to have a very big boat in the open ocean. So this is going to be easier to harvest, be easier to visually check for growth. It might just be something you could wade and walk in. And muddy shores or estuaries, again, they have more nutrients there. In the open ocean, like on the seabed, there's not as many nutrients. You don't have as much influence from like coastal runoff or things like that. Disadvantages. We're going to have exposure to air at low tides. This could cause desiccation. It could also cause predation by birds. By birds is underlined because that is the reality of what would be eating them. It wouldn't be something else that would be in the water because they're out of the water at the moment. There's less time to feed due to the tide changes, so due to tides. So therefore there would be less growth, and those go together. So suggest less time to feed, and then explain how that would matter or how that is a disadvantage. There would be less growth. Because there is less growth, we would have a longer time to harvest, longer time to get them to sell. If we have excessive runoff from the coastline, or from coastal areas, coastal communities, runoff with sediments could clog the gills. And because, again, you're near the coast, you're more susceptible, so higher susceptibility to algal blooms from having those excess nutrients, or even pollutants coming through runoff. Because again, you're near the coast, you're in an estuary. All right, we're almost done. Then we're in section B, and that's that. So we got just 4 pages left. Looking at that, an atom of oxygen, and it says complete figure 4.1 to show a molecule of oxygen O2. I have some thoughts on this, of course. This is not stated in your syllabus to be able to do this. This is, again, more of like an AS level syllabus question. But oxygen has 6 valence electrons, and that's an awful circle. It has 6 valence electrons. It would like to have 2 more so that it could have 8, and that is your octet rule. And having 8 would allow it to be in equilibrium. And now, honestly, because this is the first time this question is on the syllabus or even the AS level syllabus, this question is the first of its kind. I'm just going to follow the pattern that they did initially. And we just want to show that there's the octet rule, which means 8 electrons in the outer shell. And if you've done any chemistry, or again from AS level, oxygen has a double bond. So when you write it, it looks like this. And so that means here's an electron being shared, an electron being shared, an electron being shared, electron being shared. Ultimately, one atom of oxygen is sharing 2 electrons, and this one over here is also sharing 2 electrons. And if I just go through and count them really quick, you can see why. We're not going to focus on this first the first orbital, this one right here. That orbital can only hold 2, and it is stable at this moment. But on the outer shell, we have 1, 2, 3, 4, 5, and 6. So it's going to require 2 more, and it's going to get those 2 more from this atom of oxygen we just drew in. And I am just because, I haven't seen this question before, I'm just going to duplicate what they have on theirs and then add what they wanted. And so oxygen has 6 valence electrons. Valence is outer shell. 1, 2, 3, 4, 5, and 6 are going to go right here. That's where they're sharing. And I'll actually do them a different color just so you can see what I'm talking about. Do not color anything on your exam at all. It will not show up the way you want it. And so now this one has 1, 2, 3, 4, 5, 6, but this is the 7 and this is the 8. So now the atom of oxygen that I just drew in is stable. And in the one that I drew before, the one that was drawn for you before. You have 1, 2, 3, 4, 5, 6, and this again is your 7, 8. Nope, might be 7, 8. So they're all sharing together. You don't need to count them. The way to get marks on this, and again, you would do this in your pen, or you could do it in pencil first. Marks is that you share electrons, and then you need 2 sets being shared, or two sets, 2 pairs shared. And so here's one set being shared, here's the other set being shared. Studied by the conservation group, the IUCN, the International Union for Conservation of Nature, has found, and this is also people who have the red list, and I did that 2 videos ago on 3/1, May, June 3/1, has found that around 700 ocean sites are now suffering from low oxygen levels compared with 45 that was in 1960. The amount of oxygen dissolved, the concentration, or the amount of oxygen dissolved in oceans has declined globally by an average of 2%. However, in some tropical locations, that's a hint, it's declined by 40. So tropical, we need to be thinking high temp. So what's going on that's different? This is all about oxygen or gas solubility. Some scientists think that climate change is one of the main causes of this decline. And we're talking about oxygen in the oceans. Explain how climate change could cause the decline of oxygen levels. This is along the same lines of some of the evidence tried to refute that humans cause global warming. Is that, well, it doesn't necessarily have to be that there's more CO2 in the atmosphere making the oceans warmer, the atmosphere warmer, causing the oceans to be warmer. What if it's that the oceans are warmer and then there's low gas solubility in the ocean, therefore, and it's going to cause that CO2 that is in the ocean to be released into the atmosphere. So what if one's causing the other? It's very much along those lines right here. Okay, but how could climate change cause this? All right, Earth's atmosphere is getting warmer. Oceans absorb the heat. And then there's a couple ways that this could cause less oxygen. So we could say O2 is less soluble. In warm water. Additionally, we could say that warmer water has more evaporation. Which would cause an increase in salinity. And then with that, oxygen is, going back to this mark right here, oxygen is less soluble in warm water. Also, it's less soluble in high salinity. Those all go together. And we could say warm water could denature enzymes. Of some producers. And then that would cause a decrease in photosynthesis. We're not going to put photo tomorrow. Suggest why in tropical waters, so warm, species like sharks and tuna, and hearing sharks and tuna, you should be thinking very active animals. They constantly swim, they're big, they're very active. So they require a lot of oxygen so they can do respiration. Why in tropical waters are sharks and tuna more at risk from declining oxygen levels, all about respiration, than species like jellyfish and sponges. So those are not as active organisms. They don't require as much oxygen for respiration. Sharks. I'm going to put sharkies. Sharks and tuna are larger or even better, more active animals. They move a lot. They require more energy for muscle movement or muscle contraction. ATP or energy is provided or synthesized through respiration. And we're not going to spell through like that either. It's not a drive through. We're just doing this quickly. And linking it all together, why are we talking about oxygen, or why are we talking about respiration? If the question asks about oxygen, well, aerobic, that means with oxygen, respiration requires oxygen. So you always want to geez, Lewis. You always want to link respiration with oxygen, oxygen with respiration, and then energy requirement. Sunlight and photosynthesis, dissolved organic chemicals with chemosynthesis. All right, this is a big question. And that bigger last one's done. I'm just finding some motivation to continue. Okay, we've seen a lot of these now, more so for the aquaculture questions, but talk out the advantages and disadvantages of building artificial reefs to rehabilitate depleted fish stocks. So what are the pros and what are the cons to using artificial reefs for this? Okay, should be a big one. All right, and you can do like a T-chart, like I mentioned before. And we're not going to use any color on your test. Not on your own answers. I don't like the way that looks, honestly. TBH. Why that happened, why that happened, why that happened. Yeah. You can highlight words that they give you, graphs that they give you, data that they give you, but you cannot highlight anything that you're going to be turning into them for a grade. It just won't be able to be read. Once it gets scanned through, anything that's in color will end up being like a grayscale. All right, and these will be my disadvantages. Okay. Building artificial reefs to rehab depleted fish stocks. Some advantages. This is going to provide shelter from water currents, waves, or storms. Another mark we could say is provide shelter from predators. So provide shelter. All right, and the quotes mean whatever I wrote above it, just bring it down. This can add to or enhance the existing habitat if there's already one, or provide new habitat. And habitat is underlined. That needs to be the word that's used to get that mark instead of saying places to live. It can provide a substrate for sessile organisms, so organisms that don't move. ORG is organism. Okay, and this could be producers. Algae, corals, sponges, larval attachment, filter feeders. Now, if we have an increase in producers, we'll attract more consumers. Or instead of attract, you could say can provide food for more consumers, and this will increase biodiversity because now you're going to have a variety of different species living there, and within those species you could have a lot of genetic diversity, and you are, by definition, by putting in an artificial reef, you're making a new habitat. That means you're going to be increasing the ecological biodiversity. The artificial reef will provide nursery areas. And this would allow populations to increase because you can have now their juveniles growing there. The artificial reef can also be used with aquaculture. So this doesn't necessarily have to be in the sense of we're going to try to rehab depleted fish stocks only for wild fish. We could also be doing this for aquaculture and farming fish, and at the same time be using an artificial reef to help do that as well. Because the artificial reef is going to provide all the benefits to aquaculture that it also would in a natural environment. So within that same one, it could be used with aquaculture to farm or grow fish or shellfish. Shellfish are your filter feeders. Will improve water quality on the reef. Shellfish are your filter feeders. Farming fish aside, just what about the wild populations? An increase in fish populations. So just context for this, that's why I put it in parentheses. Provide a source to harvest for fishing industries. And there may be another way that you can say that. Like there'll be an increased amount of fish to like provide income for fishing industries. And all in all, this will reduce the pressure, decrease the pressure on natural reefs. The pressure is already there from the fishing industry. Disadvantages, the material for the construction of it. You need to place this on the seabed. These are extremely heavy. You need an experienced, knowledgeable team of divers that can guide this into its exact place, and then you need to get it attached to the substrate. It's very intensive, and they're expensive. So the material used for construction of the artificial reef needs to remain on the seabed could, this is market in of itself, move during winter storms. Winter is a time of very stormy seasons in the ocean. In that movement, so we'll say which damages benthic or bottom, habitats or organisms, or damages natural reefs, unsafe materials could be used. And those can be toxic to organisms. Give an example. Ships or tanks, like military tanks or old ships, deliberately sunk, could still contain oil and paint. And some of that paint might have like TBT in it to prevent fouling. Corrosive metals, batteries, the unsuitable material. So this material could erode very quickly, and then that's such a waste of money. So the reef ball is actually better. Thus, only providing a temporary habitat. It's a waste of money, waste of resources. The cost of materials will be high. Reef balls are very expensive. They're usually on a low end average about $10,000 a reef ball. And then, you got to hire a ship that is able to carry it out to where you need it to go. There has to be anchor points so that it stays on the substrate. But the materials, the whole venture, the whole creation of it, hiring people that know what they're doing, that can, snorkel down from the surface and direct this to where it needs to go, anchoring it in, it's very expensive. But if you have to compare like the cost analysis and see what it could do to improve fish stocks, or if you're using it to prevent further erosion and have it be like something to block waves and water current energy, to stop erosion, to stop loss of coastline, then it could be worth it, but it does require a lot. It could introduce a spawning ground or a breeding ground for invasive species if there's like a new habitat available for them. SPCS is my abbreviation for species. And we got two more. It takes time, or taking years, for like that ecosystem to develop. So you gotta try and see if it's worth it to wait that long, or it might be too late. And then if you do have that new area there, the reef ball or your artificial reef, tourism Tourists like to go see where there's a lot of biodiversity, and tourism could drive away your fish stocks, your fish population. And so then what was all this for? Something else maybe that you could include for disadvantages, you probably need to introduce some legislation or some like restrictions on when people are allowed to fish there and if they are allowed to fish there and when. All right, some pumped ventilation. Ventilation for your grouper. And then, so after this page, there's just one more about the invasive species. So this is pumped ventilation. When we see the P in pump ventilation, we need to talk about pressure. And the V, we need to talk about the volume. This is also called buccal pumping. And so we're going to start this off always, especially whenever it's starting off. Sometimes it says during the outflow of water. This is ventilation in a grouper during the inflow of water. So water's moving in. The mouth opens and simultaneously the operculum closes because we need to create an area of like a vacuum. Here we go, talk about volume. The volume increases in the buccal cavity. Buccal, think like your cheeks, like, or sometimes people say buccal, like I need to do a buccal swab or a buccal swab. It is the cavity that's like, the cavity is like an open space. It's like inside of your mouth. So it's like you're opening and closing your mouth. The contraction of your jaw, that's like, those are your buccal muscles. And yeah, if they're doing like a drug test or DNA analysis, they'll swab the inside of your mouth. They call it a buccal swab or a buccal swab because that is your buccal cavity. Okay, so volume increases in the buccal cavity, or you can say inside the mouth. Due to contraction. Of buccal muscles. I have to think sometimes, how am I spelling muscles? Am I talking about the shellfish or am I talking about like a body? And you know, if you've talked all day long, I know it happens to me a lot, that my mouth is literally sore from talking, just that opening and closing, because that requires energy. That's the muscle and it contracts. So this requires ATP. This is active transport to keep contracting a muscle. ATP is always required whenever there's muscle movement or muscle contraction. Okay, so we talked about the volume and then how that happens, how the volume gets bigger. So now we have this open space with, at that exact split second moment, there's no water in it. And so the water's not in there. There's no water in the grouper's mouth because the pressure inside of it's low. Any water that was in there, it flushed over its gills and forced it out the operculum. So now, clean slate, there's nothing in the mouth. It opens its mouth and it's empty. A bunch of water is going to flow in. And that's because the water pressure specifically, water pressure in the buccal cavity or in their mouth is low. And it's probably best for you to refer to it as buccal cavity because then it maybe can hint to you to mention buccal muscles. They're not called mouth muscles. Okay, and because of this low pressure, this will cause water to flow into the mouth. And this last mark, you could actually start with it in the beginning. This is referred to as pumped ventilation. And this is when you see fish just, you know, they can be stagnant and just like staying in one area, opening and closing their mouth, they're breathing. In the last video, I mentioned my thoughts on these kinds of questions. I'm just going to take a drink of something real quick. Explain why sharks and grouper are classified in the same phylum but different classes. This is such an A-level or AS level question. And on their examiner report, students, didn't do well on it, and I'm not surprised. It's not in their syllabus. And some of these things, like what puts them in phylum Chordata, truthfully, honestly, no one's going to remember that. Maybe like one or two of those features, maybe outside of their own exam. Like, that's something that you're glad to forget. I won't forget it because as long as it's on the syllabus, it'll be my job to teach it. But that would be way too much to have people in A level be like, oh, and by the way, don't forget to brush up on the AS level topics. Like, that doesn't ever happen. I've now only seen that happening with the biodiversity question in the last paper and right now. So, whatever, we can't control it. We can just think about this is where I need to put some emphasis. And the things that you're like, that's insane, I'm not going to do that. Majority of you guys will all think that's insane, I'm not going to do that. Like, I'm not going to review the entire AS syllabus. I don't blame you. I don't blame you. And so, you know, you guys all kind of curve the test yourselves by your own successes. So I don't know, maybe internationally everybody can agree, like, I'm not covering the AS level syllabus. If I'm in the A level class, I'm not, restudying all of that. So can we just all agree to just bomb those questions? I'm just kidding, but you know what I mean. I'll go over it anyway. So they want to say why they are in the same phylum but different classes. So they're in the same phylum, they're phylum Chordata because, for the phylum, they are both chordates, which means so at some point in development, and we're also chordates too. So this could be like in our fetal stage, and other organisms could be in their larval stage. They have a notochord, a dorsal neural tube. Now we have these things, but we eventually get a spinal cord and a nerve cord. Pharyngeal slits, which, like on a fetus, looks like these. It's like the gill slits. And then a postanal tail. I mean, we all have these things too, just we grew out of it. So that's that's part of it. And then they're in different classes. I think if I'm remembering correctly, the bony fish is from Ostichthes, and then Chondrichthes is the cartilaginous. I would have to look it up, and I'm not going to. So they're both in phylum Chordata, and then like they split off to their bony fish or cartilaginous fish. All right, so sharks have, and for this question, you would need to say what sharks have and then what bony fish have. So you don't want to say sharks have gill slits, period, or sharks have gill slits, bony fish don't. Because saying that they just don't, provided there is another answer that's there, and so if they don't, then what do they have? You would need to say both. All right, sharks have a cartilaginous skeleton. It's all about the skeleton. We don't want to say bony fish don't. We would say bony fish. A bony skeleton, and it's about the skeleton. Because you wouldn't say like humans have bones, but we also have cartilage, but our skeleton is made of bone. Another thing, sharks have denticles. BF, bony fish have scales. And that's not necessarily marked enough itself. Sharks have the gill slits. I'm getting shorter and sloppier every time. Bony fish don't. You can't see them. They have the operculum. The gill cover. BF, bony fish have swim bladder. And this is the only one where we would say sharks don't. Sharks have a very oily livers, and that will allow them to be more buoyant. All right, so explain why, give reasons why invasive species, marine invasive species, are successful. So that's weird because we think of them as like something that's negative, which they are. But why are they so successful, or how do they get to be so negative in their environment? How come they're able to outcompete? How come they're able to dominate that ecosystem, essentially? And outline the negative impacts they have on marine ecosystems and the local community. So a little bit of a loaded question. Explain why, give reasons why they are successful, and outline negative impacts on the local community, but also outline negative impacts on marine ecosystems. So the way we start this out, and this now is a totally acceptable question to see on your syllabus for invasive species, we need a definition. You could always start the definition with something provided you have the definition of it, or you were taught the definition of it as part of your syllabus. And some examples. If you're not sure if you can give an example, do it anyway. Typically, examples are supported in describe or explain commands. Invasive species are non-native and cause harm to the environment. So they don't belong there, non-native, and they're not good. They cause harm. You have to have both. How do they get there? They can get released new ecosystems by escaping from aquaculture, sea cages, or released by ballast water, which is water that's discharged from ships. They could be accidentally released by humans, or they can biofoul on ships, which is why we would use anti-fouling paint. An example, the crown of thorns starfish, or a really popular one in Florida is the lionfish. Some of you guys might be familiar with the zebra mussel. That would be another one. Why are they successful? So they have a very rapid growth rate. They grow really fast. So that means their fecundity, or like their reproductive abilities, are very high. Their breeding successes, the recruitment, so the new organisms coming into the environment, all of that is very high. Invasive species can eat a wide variety of prey, so they have plenty of food items. And they have few or no natural predators in their ecosystem. So all those things, they don't have many predators to stop them. They can eat so much food, they can grow really fast, and they can reproduce and have a lot of themselves, make them very successful. Some negative impact on ecosystems. Negative impacts on marine species are they can outcompete the native species. Sounds like something that you're going to want to say anytime with invasive. This will force native species to become endangered, maybe even extinct, or they're going to have to migrate to new areas. So they're outcompeting them. And as a species, you're either going to allow yourself to be defeated and die, or try and fight it, get outcompeted, and perhaps lose, go somewhere else to migrate. And if they leave their area, this will decrease biodiversity. When you're decreasing biodiversity and you have species leaving, this can permanently alter your habitats or alter food webs. Okay, and these are going to be the negative impacts on the community. So local community is referencing like humans. What color am I not used? Perfect. Okay. Negative impacts on the local community. So human communities. Invasive species will cause money loss or income loss or economic losses. There could be employment loss, tourism loss, fishing industry money losses. This will decrease the numbers for fisheries to catch or food for humans, because again, they are eating a lot of the food sources. That's not just other organisms' food sources, but humans too. They're expensive to remove, and to start introducing prevention methods to humans to start to do, that could also be difficult. And those prevention methods typically don't always work without something else negatively occurring. They could introduce disease into the local ecosystem. And all of those things would cause there to be a loss of income for fisheries and therefore loss of employment. Anytime you're bringing in a new organism into an environment or introducing one in or mixing two species together in an environment, disease is always a wonderful thing to talk about. It is a really good go-to. Okay, so that is literally now all of the paper 3 examples that I have.